whoops. Well, okay. Okay. So there's the neutral loss that falls off, and it continues on its way. So that's the second prop here. No more props. So you can, okay. So here's the actual chemistry. We take advantage of a sulfate or a quaternary amine where the sulfate can readily lose an SO3 neutral and then becomes a phenolate anion, or it's a benzylic quaternary amine, and then we get a benzylic cation. So you see what we have here is resonance stabilization of the ion, and that ion stays with the analyte, so we get an analyte characteristic signal. And so this is the basic idea of these tags, and then they're charged up front, so they really do well in the ion source. So here's an example where one could use some more sensitivity. Let's say a woman has breast cancer, for example. There's a new case of breast cancer diagnosed in the United States every 3 minutes. 250,000 cases per year, of which 200,000 are invasive. Half of breast cancer is estrogen or steroid dependent. This estrogen as a growth signal is driving the formation of that tumor. A very tiny amount can do that, like a picogram per mil can drive that breast cancer. In one therapy, what you do is you have a drug that blocks the estradiol from its effect on that tumor. So that's one approach. In another approach, you've got therapy that blocks the synthesis of the estradiol. And there what you want to do is you want to know, have you given enough of the drug to take the estradiol down to the level where it's not driving the tumor. And so how do you do that? And so if you can't do that, then you have to wait a few months and see if the tumor is growing more, and then you step up the dose, and that's not a very good approach. So what you do is you take this serum sample and you send it to the Mayo Clinic in Minnesota, where they have set up a mass tag assay for estradiol, as illustrated here. The mass tag is a Danzel group. It has a positive charge. They react the estradiol from the serum sample with the Danzel reagent, and then when they detect this in the mass spectrometer, the Danzel falls off and carries the charge with it. And that's not where you want the charge to be, because now every steroid, every substance that you tag will have that same ion signal, which would be the tag. You want the signal to be with the estradiol. Mayo Clinic will send the sample back because their detection limit is about 5 picograms per mil. It's not enough to do those kind of analyses. The Mayo Clinic is state-of-the-art in estradiol technology. They get academic clinical laboratories or hospital laboratories will be giving them $5 to $10 million a year in business. They're a major reference testing laboratory, and that assay is beyond their scope for those kind of low levels of estradiol. We have developed an intensified neutral loss tag for estradiol, and here we see on the right-hand side we have 2 ions that are characteristic of the estradiol, and our sensitivity is about 1,000-fold more than what the Mayo Clinic has with their Danzel tag, and so this is an assay that we're moving towards application. In the bigger field of clinical immunodiagnostics, the market is about $6 billion per year currently. It should reach about $10 billion by 2014. In terms of estradiol tests, there are 3.7 million tests per year. Labs charge $150 to $250 for one of these tests. There are a lot of discounts, but at any rate, the market for estradiol could be, maybe $80, $100 million a year. And so these there's a lot of interest in that kind of analysis. There are also a lot of opportunities, for example, testing mouse samples where you don't get much serum and you can't reach those samples with some of the existing technology. So Pat Sluss, the director of chemistry at Mass General Hospital, says the immunoassays cannot survive. We need more sensitive, accurate, multi-analyte analytical technology. The answer is mass spec, if only it was more sensitive. Paul Taylor in Australia says, okay, if we're going to bring the mass spectrometer into the clinical laboratory, we need to make a quantum jump in the technology, like MALDI, for example, and to combine that with like an antibody kind of cleanup techniques to get the throughput you need. And we have submitted a patent application on antibody technology with mass tags. So let me jump to two other application areas very briefly. The second example of chemicals where one wants very high sensitivity is detecting chemicals attached to DNA. Damage to DNA is the basis of all cancer. Chemicals can attach to DNA. They, like gum, gets stuck in your hair. It's covalent attachment. The name for that is DNA addicts. Those can go on to mutations. Of all the substances classified by the International Agency for Research on Cancer as carcinogens, in nearly every case they're known to form DNA addicts, and they're of interest in other diseases, preterm birth, for example. So addicts are present at very tiny amounts, and so one application area is drug development, because the FDA is very concerned about genotoxicity, as is the drug companies. And then the dream is a cholesterol test for cancer. When you get your cholesterol's elevated, the doctor gives you advice about what to do. You already know a lot of the advice. How about a corresponding test for cancer to know what chemicals are on your DNA and get some specific advice about what you should do about your diet, lifestyle, occupation, whatever, to potentially bring down your cancer risk. So that's kind of a dream for this. So this is what might be in the future, is to get a profile and then fix your profile and reduce your cancer risk. The idea has been around for 40 years. Some ideas are before their time. There's the idea of walking on Mars, that hasn't arrived yet. So this idea has been around a long time, but the technology has not been there. So the mass tag that we have in this area is a benzoyl histamine that came out of a synthesis program, and here it's attached to a phosphate of a nucleotide of DNA. This is a model DNA addict where the acetylminofluorine is clunked on here, and it's not supposed to be there. This would be a normal nucleotide if it weren't for that, and then we put our mass tag on here. And so example of the application of this assay is we're doing some testing of genotoxicity for Pfizer. They're sending us animal samples and cell culture samples, and they're saying what chemical would we use as an exposure here, or how about some quantitation, and then we've been giving data as seen here. And the question is, why don't they do this in-house? They tried to set up an assay in-house, and it wasn't successful. And why aren't they sending it elsewhere? And we have the only assay in the world that can detect DNA addicts broadly with high sensitivity, so we're very, very proud of that, obviously. We have some collaborations underway with this. One application area is the preterm birth. This one is from an investigator at the University of Turin, and this was a project here is exposure of pathologists with formaldehyde, and then we did some DNA addict profiling of some standard samples. So in regard to preterm birth, we have this program project grant with the Crown as the PI at Northeastern, and he's the PI with the overall project. It's a $26 billion burden to society, and it's been going up, and especially in Puerto Rico, a lot of Superfund sites there. And so it looks like it's environmental contamination, and a mechanism of that may be damage to DNA at the fetus stage, for example. And then last is an example of an environmental analysis. You've heard about bisphenol A in plastics, and great concern about this in legislation. And so here we have a mass tag where if we're going after an easier analyte, like this is a picomole, it's an easier analyte, then you get a different advantage, and then you get an advantage where you don't need to do any cleanup. You simply label the sample and put it right in the mass spectrometer, and so that's what we did here. So as I said, the impact is this early kind of detection that can help you in a number of areas. Okay, thanks.
It's great to be back. This is my alma mater. Did my PhD here under Roger many years ago. Let me kind of start off by saying one of the first projects I did in the industry was Estradiol. And Roger just showed us some data measuring estradiol at ultra-sensitive levels. But back in the day, we'd do it with radioactivity in antibodies. And there's a revolution going on, a very slow one, by the way, of moving away from antibodies to mass spectrometry. And think about it, the assays that are done by antibodies today, it's in the hundreds of millions per year. And it adds up to billions of dollars of revenue. And it's very interesting watching this happen. You know, my career is about product development. But there's the mass spec suppliers, and we have some here locally in the Boston area. Then you have your diagnostic companies, and it's all really being driven by the improvements around the sample prep. What goes into the mass spec is very clean and very sensitive, and what's coming out is new diagnostic assays. So, as a realist, you know, how long is this going to take? This could be, it could be 5 years, it could be 20 years, but I would predict in some period of time, most of the assays that are done are going to be done with the mass spectrometer. So there's tremendous opportunity to think about how to do that. And so some real data points. 2 mass spec companies just did deals, and I won't name the names, but they each sold 100 mass spectrometers for one application. So a mass spectrometer can be $200,000, it could be $300,000. Do the math. It's a lot of business. And they're not even in the diagnostic domain. They're just supplying the equipment to do it. So there's a lot of money at stake here. But to be successful, it's just not the ability to detect these molecules. It's the cost, it's the regulatory path, and it's the performance. So what are some of the issues to think about? And the first one I would say is the technical issues. You have to have a very clean sample, and Roger just showed us ways to get to the sensitivity you need. But these biological samples are very, very difficult to deal with. And there's tremendous opportunities from an engineering and biology perspective to make them cleaner and better. You make the samples cleaner, you'll get better results with your mass spectrometer. The biology is incredibly, incredibly difficult. It's still easier to put a man on the moon, and perhaps even Mars, than to understand what the molecules are in our bloodstream. And I was at the cancer meeting this past spring. It's as difficult to develop a cancer biomarker as it is to develop a cancer drug. And people don't understand that. They think it's easy to be able to detect something in blood and relate it to a disease. It's just not that way. There's about 20 cancer markers that have been developed, and it's taken about 20 years to get them developed. So we have a lot to do and a lot to learn there. Don't forget about cost. PSA, which is a classic cancer marker, is reimbursed at $29 a test. Roger mentioned $150, I think, for an estradiol assay. Anything and everything we do here is driven by the cost. You can't take an expensive assay to market no matter how good it is. So driving costs down each step of the way is very important. And kind of the last hurdle, which is often overlooked even at this stage, but you shouldn't, is the regulatory path. And it's easy to think that you've got the best assay. It's incredibly difficult to take that assay and convince a regulator who is not going to accept any risk that that assay works. And it even gets more complicated when we talk about multiplexing. I am a big fan of multiplexing. I think it's going to be the wave of the future. We'll stick to my favorite here, PSA. Right now, PSA is the test. 5 years from now, there's going to be a panel of assays. But then you think about the regulatory hurdles to get those panels of assays accepted. It's very, very difficult. And you're just going to dial that into your planning. So my whole message here is that, yes, mass spectrometry, as Roger just showed us, is the tool of the future. And really where the technical opportunities are really on the biology side and on the driving the costs down. So if you can do multiplex assays and you can keep those costs down at a very low level, then perhaps in 5 or 15 years, somewhere between that period, you'll have hundreds of millions of assays done by mass spectrometry. And that way you'll see this whole technology revolution really realized right in front of us. So congratulations, Roger, on the mass tags. I think for those of you who don't realize the sensitivity he's got down, nobody else can get down to those levels of detection for estradiol. So it is quite an accomplishment to get there. Thank you.